Welcome to Rashmi's classes. Let's get started. Answer questions for Class 11 Biology. Chapter 06 Anatomy of Flowering Plants. State the position and role of various kinds of meristems. Answer: Meristem refers to meristematic tissues, which consist of actively dividing cells. Based on their position in the plant body, meristems are of three types. They are given below. Apical meristem: it is present at the apices of shoot and root and is accountable for the rise and length. Intercalary meristem: it is present at the bases of leaves on top of the nodes or under the nodes, and is accountable for the elongation of the organcer. Lateral meristem: it is present on the lateral side and is accountable for the rise in diameter or girth. Fig. Diagram showing different types of meristems. Cork cambium forms tissues that form the cork. Do you agree with this statement? Explain. Answer: yes, I agree with this statement. Cork cambium, also called phellogen, cuts off cells both on its outer side and inner side. The cells cut off on the outer side form the cork, also called phellum, and cells cut off on the inner side form the secondary cortex, also called phelloderm. The cells of the cork are dead while those of the secondary cortex is alive. Phelloderm, phellogen, and phellum are collectively known as periderm. Describe the entire process of secondary development in the stems of woody angiosperms using a schematic representation. What is its importance? Answer: secondary growth refers to the formation of secondary tissues that leads to an increase in girth or width of dicot stems due to the activity of the cambium and cork cambium. Secondary tissues are established by two kinds of lateral meristems, vascular cambium and cork cambium. The vascular cambium produces secondary vascular tissues, whereas cork cambium forms periderm. The vascular bundles in the dicot stem are conjoint, collateral, open, and are arranged in a ring. The cambium present among the phloem and xylem in vascular bundles is known as intrafascicular or fascicular cambium. Besides this, some cells of medullary rays also turn into secondarily meristematic, and this is known as interfascicular cambium. Both these cambia collectively constitute a complete cambial ring. This ring of vascular cambium divides the periclinal to cut off cells both on the inner side and outer side. The cells cut off on the external side is the secondary phloem and the internal side is the secondary xylem. The amount of secondary xylem cut off is more than secondary phloem, and therefore, with the development of secondary tissue, a rise in diameter or girth occurs. The structure of the secondary xylem and secondary phloem is the same as that of the primary phloem and primary xylem. With the rise in secondary tissue, the primary phloem and primary xylem get crushed. The ray initials of the vascular cambium ring are split by tangential divisions and add new cells. These new cells formed on both the sides of ray initials remain meristematic for some time and then distinguish into parenchymatous cells of rays. The rays, produced by vascular cambium among the secondary phloem and secondary xylem, are known as secondary medullary rays. They are generally 1 to few layers in thickness and 1 to numerous layers in height. The medullary rays form the radial system accountable for the radial conduction of solutes. They sustain the link between the cortex and pith. There is a significant difference in the activity of cambium with a change in season. In spring, the activity of cambium is even greater, and therefore the wood elements are bigger in size, with a wide lumen. The activity of the cambium is less in the autumn and the wood components are relatively small in size, with a thin lumen. Autumn wood and spring wood of a year form the annual ring. The age of a tree can be defined by calculating the annual rings. Numerous annual rings are consistent with the age of a tree. Phellogen cells, cork cambium, split on both the inner side and the outer side as well to form secondary tissues. The secondary tissue developed on the inner side is known as the secondary cortex, whereas the tissue developed on the outer side is known as cork. Fig. Diagram indicating secondary growth in dicot stems. Significance of secondary growth is as follows. It adds to the girth of the plant, thus provides support to the increasing weight of aerial parts due to growth. It produces a corky bark around the tree trunk that protects the interior from abrasion, heat, cold, and infection. It adds new vascular tissues for replacing old non-functioning one as well as for meeting increased demand for long-distance transport of sap and organic nutrients. Draw images to sketch out the anatomical variation between monocot root and dicot root. Answer: variations between monocot root and dicot root are shown in the following picture and table. Fig. Comparative pictures of dicot root and monocot root TS. Features: monocot root, dicot root. Cortex. Comparatively very wide. Endodermis. Less thickened and Casparian strips are more important. Later turn highly thickened. Casparian strips are noticeable only in young root. Passage cells. Generally absent. Usually occur opposite the protoxylem point. Pericycle. Generates lateral roots, cork cambium, and part of the vascular cambium. Creates lateral roots only. Vascular bundles. 2 to 5 or sometimes 8. 8 or more numbers. Pith. Only. Either absent or extremely small. Well developed. Monocot stem and dicot stem. Answer: variations between monocot and dicot stems are shown in the following illustration. Fig. Comparative pictures of dicot root and monocot stem TS. Features: monocotyledonous stem, dicotyledonous stem. Vascular bundles. Scattered. Conjoint, collateral. Bundle sheath is usually present. Phloem parenchyma. Vascular bundles in the ring. Conjoint, collateral or bicollateral and open. Bundle sheath absent. Phloem parenchyma present. Absent. Xylem vessels are arranged either in a Y or V-shaped manner. Not arranged in V or Y-shaped manner. Pith, medulla. Absent. Composed of parenchymatous cells found in the center of the stem. Ground tissue. Ground tissue is not distinguished into the pith and cortex. Distinguished into the pith and cortex. Hypodermis. Usually. Sclerenchymatous. Collenchymatous. Endodermis. Absent. One-layered, starchy sheath is generally not well distinguished. Pericycle. Absent. Composed of one or several. From your school garden, cut down a T-or-answer section of the young stem of a plant and examine it by using the microscope. How would you determine whether it is a monocot stem or a dicot stem? Give reasons. Answer. Looking through the microscope, I will check if the following features are observed. Vascular bundles in the dicot stem are arranged in a ring, whereas in monocot stem vascular bundles are scattered throughout the ground tissue. Based on the arrangement of vascular bundles, it can be ascertained whether the young stem is dicot or monocot. In addition to the indistinguishable ground tissue, sclerenchymatous hypodermis, spherical or egg-shaped vascular bundles with Y-shaped xylem are other distinguishing characteristics of monocot stem. Fig. Arrangement of vascular bundles in dicot and monocot stems. The T-or-answer section of a plant material reveals the subsequent anatomical features. A. The vascular bundles are conjoint, widely dispersed, and encircled by a sclerenchymatous bundle sheath. B. Phloem parenchyma is absent. What will you find it as? Answer. The plant material is identified as a monocot stem. Why are phloem and xylem known as complex tissues? Answer. Complex tissues are made up of more than one type of cell, and these work together as a unit. Xylem elements are responsible for the conduction of water and mineral salts from the roots to the other parts of the plant. Xylem elements are highly lignified and dead except xylem parenchyma. It consists of xylem vessels, xylem tracheids, xylem fibers, xylem parenchyma. Fig. Complex tissue, xylem. Phloem is a complex tissue associated with the T-or-answer location of food. Phloem elements are living except phloem fibers. It consists of sieve tubes, companion cells, phloem fibers, phloem parenchyma. Fig. Complex tissue, phloem. What is the stomatal apparatus? With the help of diagrammatic representation, describe the structure of stomata and label its parts. Answer. The stomatal apparatus comprises the following parts. A small aperture or opening present in the epidermal cells of the leaf called stoma. This is also called a stomatal aperture, singular, stoma, plural, stomata. Two bean-shaped guard cells surrounding the stomatal aperture. It is to be noted that guard cells are dumbbell-shaped in monocots and bean-shaped in dicots. Subsidiary cells, these are specific epidermal cells in the vicinity of guard cells. Function. Change in the turgidity or flaccidity of the guard cells is associated with stomatal opening and closure. Stomata are essentially involved in gaseous exchange and T-or-answer. Fig. Stomatal apparatus. Mention the three fundamental tissue systems in the flowering plants. Provide the tissue names under each system. Answer. The three basic tissue systems in flowering plants are the epidermal tissue system, ground tissue system, and vascular tissue system. The epidermal tissue system comprises epidermal cells, stomata, trichomes, and hairs. The ground tissue system is composed of the endodermis, cortex, pith, pericycle, and medullary rays in the primary stems and roots. In leaves, the ground tissue is comprised of thin-walled chloroplast which contains the cells and is known as mesophyll. The vascular tissue system is composed of complex tissues, the xylem and the phloem. In what way is the study of plant anatomy beneficial to us? Answer. The study of plant anatomy helps to understand structural adaptations in plants with respect to their different environmental conditions. It also helps us in differentiating between monocots, dicots, and gymnosperms. This gives us an idea of the physiological state of the plants and so can be useful in crop improvement. Internal structures also help us to predict the strength of wood and hence its utility for commercial activities. Study of plant fibers such as jute, flax, and hemp, etc. may prove useful in their business-related exploitation. What is periderm? How does periderm development occur in the dicot stems? Answer. Periderm is a protective layer present outside the stem that replaces the epidermal layer in response to any injury or invasion of pathogens. Phyloderm, phylogen, and phloem together constitute the periderm. Dicot stems produce phylogen or cork cambium in the external cortical cells. Phylogen cells split on both the inner side and the outer side to form secondary tissues. The secondary tissue produced on the inner side of the phylogen is known as the phyloderm or secondary cortex. On the outer side, phylogen produces phloem or cork. Define the inner structure of a dorsiventral leaf by using a labeled diagram. Answer. Fig. Internal structure of a dorsiventral leaf. Dorsiventral leaves are found in dicots. The significant anatomical characteristics of dorsiventral leaves are discussed below. Upper epidermis. This is usually the outermost single made of parenchymatous cells. The epidermal cells have sometimes outgrowths known as papillae, example, in gladiolus. The epidermal cells are devoid of chloroplast and stomata are absent on the top epidermis. Lower epidermis, it is the same as the upper epidermis, but here stomata are present. Chloroplasts are absent in the bottom epidermis also, except the guard cells of stomata. Mesophyll, in between the lower and upper epidermis mesophyll tissues is present, which can be split into two regions. Palisade parenchyma, these are lengthened columnar cells, without intercellular spaces. These contain chloroplast in them and are usually arranged in two layers. Spongy parenchyma. It is found below palisade parenchyma and is oval or spherical with intercellular spaces. They also contain chloroplasts, but several chloroplasts are more in palisade parenchyma than spongy parenchyma. Vascular bundles. Vascular bundles are usually found at the boundary between the spongy and the palisade regions. The vascular bundle in the midrib area is the largest. Vascular bundles are collateral, conjoint, and closed. Each vascular bundle is encircled by a bundle sheath of parenchymatous cells. In the vascular bundle, phloem is found towards the lower epidermis and the xylem is present towards the top epidermis. Additionally, in the xylem, the protoxylem is towards the uppermost epidermis. Thank you for watching. Please like, share, and subscribe.